Hi,
Welcome,
everyone. Hi.
welcome. This is John Bagnulo, and I'm really excited to talk to you today about the importance of protein quality and how it is that protein quality is assessed in different foods, as well as the value that specific amino acids, essential amino acids, bring to our lives, to health, and especially to patient health. So with that, let's get started. This is a topic that and I guess I would first say, complete disclosure, I serve as director of nutrition for nutritional medicinals. My learning objectives for all participants today would be that you can identify three different methods of assessing protein quality, describe those, as well as identify populations where consuming all essential amino acids in a very small window of time, i.e. the same meal, is required for optimal growth, recovery, and improving outcomes, as well as describe two clinical signs of inadequate or suboptimal essential amino acid intake. And lastly, to explain how three different amino acids signal metabolic change or play important physiological roles for our patients. And this topic is so important for many reasons. I think one of the most fundamental reasons is people are always interested, both clinicians as well as consumers. They're always interested as to what the difference in protein quality is from one food to the next, especially animal versus plant protein. I think that's a real central theme in this conversation. And there are so many questions that exist around what is optimal. What are the optimal levels of specific essential amino acids as they relate to human physiology, as opposed to a broader mammalian model, or maybe looking at the rat model or the pig model. And I bring these up because historically, rats and pigs have both been used as surrogate models for protein metabolism, as research has always tried to look at growth rates and how different foods affect muscle synthesis and weight gain in different animal models. So, it's really fascinating to think that so much of our calculations with respect to protein quality are based on these other animal models. We'll take a look at that in a moment. But these are some other important questions that I think we all really need to ask ourselves. Is there a particular type of protein quality assessment that best reflects human needs and a human's ability to assimilate protein? And then we could expand upon that and say, is there a protein quality assessment that's most useful for, let's say, a critically ill patient? Is there a single plant food that can, match the protein quality and effectiveness of a animal source of protein? Do these incomplete proteins that are giving us, different levels, or they have varying compositions of essential amino acids, do they need to be combined at the same time? And again, are there specific amino acids that might be more influential than other essential amino acids in determining the recovery from a specific condition, or something like the loss of lean body mass in certain patient populations? So with those questions and those learning objectives in mind, I just want to refresh everybody's memory for one of the most fundamental points here today. Essential amino acids are broken down into really two groups. We have the true essential amino acids, those 8, which are needed throughout the lifespan by all populations, all human populations. And then we have 4 that are considered conditionally, and sometimes histidine is put there as well as a conditionally essential amino acid. So histidine has that's in that gray area, but arginine is definitely considered the most classic of the conditionally essential amino acids, meaning that it is needed at certain times of development and in certain conditions for humans to adequately grow, recover, gain lean body mass. And then the others are the non-essential amino acids. And the non-essential, would certainly make it sound as though that they weren't important, but they do obviously play very important roles in our physiology, and we could talk extensively about all of these, especially glycine, for instance, which is very similar to methionine, which we'll talk about in its role of methylation in the body. But these are our essential, conditionally essential, and non-essential amino acids. Now, when foods are assessed for their protein quality, there are several different methods of determining protein quality that we're going to look at. First, and this was the first one really used historically, it's the protein efficiency ratio. And, really how that was determined was looking at animals, and it was rats primarily, looking at the growth of an animal, primarily rodents for the purpose of research and the ease of studying them, how they grew and the amount of weight that they gained in relationship to any particular food that was being consumed over a test period. And, again, it's very rudimentary, but that was kind of the foundation for some of these others going forward. Now, a non-growth, I guess you'd say growth-related test or measurement is what a lot of us are familiar with, which is the amino acid profile or the chemical score of a protein. And that looks at the milligrams of essential amino acids that are found in every gram of protein that comes from any particular protein source. And then it's always compared against the egg. Typically the egg, which has a 1.0 or a perfect amino acid score. This is very useful. Again, I don't want to in any way diminish the usefulness of this, because it isn't, what we're going to talk about here in a few moments, about some of the more contemporary methodologies of looking at protein quality. This is very useful because it identifies what we refer to as rate-limiting amino acids, which are those amino acids that are not present in adequate amounts to support optimal or normal growth, weight gain, muscle, protein synthesis, etc. Then other little slightly more complicated methods of protein quality that are, again, I think these are they still have great value. And one of those is biological value, which is looking at the proportion of absorbed protein from any given feed or combination of foods, but from a protein source that is incorporated into an animal's body. Again, I want to emphasize the fact that humans have rarely been used for these methods of assessment. These have almost always relied on other mammalian models. Rats and pigs are the most common models, and still are to date for some of what we're going to talk about. But this biological value is important because it's looking at how much of a protein consumed is absorbed, which then the assumption is if it's absorbed, then it must be used by the body for protein synthesis. And then we know that's not always the case. So every one of these will have limitations, from the theoretical limitation of the amino acid profile, the chemical score, meaning that it may have high levels of essential amino acids per gram of protein. However, does that equate to weight gain or growth or recovery? The answer obviously is not always. Has to be digestible, has to have a certain level of bioavailability. And then we get into these other tests, or methods of assessing protein quality. And again, there are assumptions being made with all of these, and that's what I'm trying to emphasize here today. And net protein utilization, again, just as it sounds, it's looking at the amount of amino acids that are converted to proteins or retained in the body in comparison to those supplied by the diet. So very, very similar to biological value. And this would be, just quick illustrations historically going back to their origins. You look at the World Health Organization's, first development for net protein utilization, and it showed that in comparison to an egg, which had a score of 100, that's part of the reason why, eggs are still looked at when it comes to this chemical score that we have for foods as a 1.0. Egg has always been considered the kind of the gold standard, so to speak. But something like fish has a net protein utilization of 83. And a lot of that has to do with its limitations with respect to tryptophan. And then just a basic, just a basic equation for the protein efficiency ratio, looking at weight gain in relationship to protein consumed in grams, and, has a factor of 100. It's just a real simple calculation. Now, here is one of my favorite charts, which I've always used in teaching this to nutrition courses at universities, which is when you take a look at biological value, you can also at the same time use the chemical or amino acid profile, chemical score, and you can determine with the biological value the role of rate limiting amino acids, which is very useful again for combining protein sources to provide a higher biological value, or in essence, more complete, more effective protein source. And so you can see that, for instance, here we have cow's milk, and it has, we're looking at a 0.7 for methionine and cysteine, your sulfur-containing amino acids. So that's why if you scroll down and you look at other sources of methionine and cysteine, you can see why cow's milk combined with corn, which is at the bottom, which has a 1.1 for methionine and cysteine, would provide a very high biological value. So again, this is looking at the combination of biological value as well as the rate limiting amino acids and the chemical score to really provide a food manufacturer or a clinician or an individual that's really just trying to, create the highest quality protein in their diet, giving them as much information as possible to put the pieces together. And this is looking at the biological value in a different way. This is looking at the biological value of squid, a type of seafood. It does seem very esoteric, but I thought it was important just to show that you can basically determine this biological value for just about any food you can imagine. You look at the essential amino acids that are included in comparison to the World Health Organization, ideal or standard value, and then you can look at every essential amino acid in comparison, and then ultimately determine that biological value when you also concomitantly measure, the amount of those proteins that are retained in the body of an animal consuming them. So now we'll shift gears into three of the newer methods. I say newer, last few decades, with the exception of the last one, the digestible indispensable amino acid score, which we'll talk about here in a few moments. But nitrogen balance is something that's been used in clinical research, especially for the critically ill. But it's been really looked at as how does a particular protein source or food or combination of foods, how does that provide, let's say, whether it's an animal or a patient or any population, how does it provide them with protein to meet all of their requirements? And it's measured basically nitrogen in versus nitrogen out. And when I say nitrogen out, so many different avenues of nitrogen loss have to be captured and measured. We lose nitrogen on the surfaces of our skin from skin cells that slough off, from hair loss, as well as through what we're going to lose through perspiration and obviously fecal losses. And these studies have given us a great understanding of how protein is used and how effective it is at kind of meeting the demands of a patient or a population. Now, in these types of studies, nitrogen balance studies, every potential source of nitrogen loss has to be captured. The water that an individual would shower with was collected, and the nitrogen was therefore collected from that. But it's a part of our better understanding how particular food sources meet needs and help maintain nitrogen balance, which means would help someone maintain weight. Obviously, if someone were in a positive place here with respect to nitrogen retention, then they would be gaining weight. But nitrogen balance is looking at really the amount of protein required to meet all physiological needs and to compare intake versus losses and output. Now, these next two are the ones that are going to probably be most relevant to us today and to helping, our patients understand, what are their better choices for protein, what do they need to look at, and that's the protein digestibility corrected amino acid score, which really compares the intake of amino acids to the fecal losses, those which are not absorbed and have low bioavailability. Again, this was looked at as the gold standard, and to some extent, I guess it still is with respect to evaluating a food's ability to contribute to a person's total protein pool. It does make assumptions. It makes several assumptions. One is that a protein being consumed by one person is going to be, as effectively digested as another. We know that's certainly not the case. We know that stomach acid and pancreatic enzyme levels play big roles in that, as well as what's going on with respect to the small intestinal wall and the absorption surface area there. And, the other part of this PDCAAS, which is, I think, and led to this digestible, indispensable amino acid score, the kind of development of that is it also is looking at fecal losses as opposed to how much of these amino acids are present at the very end of the ileum. And the reason that's relevant to this is, amino acids are absorbed for protein synthesis in the body and for meeting the body's total protein requirements. Those amino acids are absorbed in the small intestine. Amino acids that survive the small intestine, when I say survive, that are not absorbed in the small intestine and make their way into the colon, well, those amino acids are not going to be used by the body for protein synthesis. Those may be consumed by intestinal or gut flora, microbes in the colon. They may be oxidized and converted into other substrates. So that's a limitation of the PDCAAS, is it's not really taking into account the fact that some amino acids may, they may not be absorbed in the small intestine, but something may happen to them in the large intestine, and they're not going to be measured in the fecal, those fecal losses, but they're also at the same time not going to help the individual meet their protein needs on a physiological or protein synthesis type basis. So again, that led to the development of this newest assessment of protein quality, which is the digestible, indispensable amino acid score. And that looks at the loss of amino acids, how comparing amino acid intake for any food and comparing what is then present at the very distal end of the ileum. And this is all done on the porcine or a pig model. The data is not complete for this, meaning that we have a large, number of foods where we don't have a score for this yet because they have not been measured or calculated when consumed by a pig and then measured at that distal end of the ileum. So it doesn't have the type of complete data on digestibility and recovery that we have for PDCAAS. But again, it appears to be favorable in a couple different ways. Number one is it can be used across different age groups over the course of a human lifespan. So as you'll see in a moment, we've got it for ages 0 to 1, 1 to 3, etc. Very, very specific target age groups with which calculations are made in terms of physiological needs, and then looking at how much is actually absorbable or has bioavailability in the small intestine. And I think that's a big part of the upside to potentially using this as well, is that it's not going to look at fecal losses. It's going to look at what's present after normal sites for amino acid absorption. So that's the DIAAS. And if we take a look at these in comparison to one another, the first thing you'll see is that, complete, incomplete, those words are thrown around a lot, but if you compare a complete source of let's say a plant protein or even something like chicken, they may be considered complete, but have a PDCAAS, which is less than 1.0. I think that's important because it may be complete, as we talked about earlier. It might have all the essential amino acids, but it may not have the same level of digestibility as another protein, where in the end, you have to be able to absorb the amino acids for them to actually help meet the body's requirements. So, you really need to have digestibility taken into account. I think that goes without saying. And, this PDCAAS can then be used to combine. This is a great, I think a great illustration of how different plant sources of protein with different levels of digestibility, and obviously having different amino acid profiles, can be used to complement one another to produce varying levels of PDCAAS when pooled together. A great example is quinoa and garbanzo beans or chickpeas. You can get a PDCAAS of 0.73 just with those two plant ingredients alone. That's not including something else such as rice, which could certainly bring it to a PDCAAS of 1.0. So you can see that PDCAAS has value for several different reasons, because digestibility is taken into account along with the amino acid content. Now, again, historically, the last couple decades, PDCAAS has been used in combination with things like the overall protein concentration of a food, as well as the allergen content. We know that certain foods might have very high PDCAAS scores or values, but they may have very high allergen content or potential reactions. Dairy, obviously a great example of that. Soy, a lot of people have concerns around soy allergies, again, justifiably so. But even though something might have a high PDCAAS, in the case of casein or soy protein, it has a high PDCAAS, it has an excellent, protein concentration. However, because it's a, real common source of allergens, it kind of fails in that area. And I feel like, we all have a good understanding of this, that we're really looking for protein sources that meet all three criteria. And then when we compare the biological value, which I mentioned earlier, and I do think that it does have still great clinical relevance. When we compare the biological value, which, keep in mind, is measuring how much of a particular protein is retained in the body and how that relates to growth of an animal, normal growth and development, you can see that some foods have biological values which are much greater than their PDCAAS score, and vice versa, of course. So, but in the case of something like whey protein, you can see it has a biological value of 100, PDCAAS of 1.0. Same is true with the egg, but where the differences arise are things with, like, let's say beef. Beef has a, biological value of 80, and it has a, PDCAAS of 0.92. So significant difference there, when you really start to compare these two. And we can compare more. We have the biological value versus net protein utilization versus PDCAAS, protein efficiency ratio, and then, points of emphasis on the far side. And you can see that there are just really, significant variations here of these scores. Something like whey protein isolate has obviously very high protein efficiency ratio, often considered to be the gold standard now, along with the egg, mostly because of its, we'll talk about this in a moment, but the role of specific branch chain amino acids in really promoting higher protein effectiveness in terms of supporting growth, weight gain. But an egg still considered, the highest net protein utilization. You'll see here high PDCAAS, very high PER, high biological value, but the biological value is not quite what you'll see in whey protein isolate. And then when you get down into plant sources, you can see that with rice and hemp and pea protein, that they have the lower levels of biological value across the board with all of these calculations. And that's why, of course, they need to be combined. But when combined, as we all know, plant proteins have the ability to be just as effective in supporting growth as any of these animal proteins. Now, as I mentioned, the digestible indispensable amino acid score, which is very new. We have a limited data set on these, but you can see that for each age group, you have different DIAAS scores for each food. And that has a lot to do with the specific requirements of an age group. And we're looking at young children here, and you can see that something like corn on the far left never even reaches the 50% DIAAS level. And things have to get over 75% to be considered good, and over 100% to be considered excellent, and would stand alone. What's fascinating is the potato. You can see that the potato has an excellent digestible indispensable amino acid score, which really surprises people. And a lot of that has to do with just how digestible the protein is. Now, obviously, we recognize the limitations of the potato, which is its protein concentration. It is so low that you have to eat such large quantities of potato to get the protein. It becomes largely irrelevant. That's why potato is an excellent complementary plant protein, but doesn't really do well on its own, simply because while it has a large number of the essential amino acids and it is very digestible, number one, it's not complete. Number two, it just has such a low protein concentration. And then right next to it, you see gelatin, which as an animal protein, I think a lot of people are under the impression that collagen and gelatin are excellent sources of essential amino acids. And in fact, they just are not. Gelatin and collagen have very, very low biological values, very low, however you want to measure them, just, missing a large number of essential amino acids and very difficult to digest until it is put into a peptide form. Then the digestibility improves, yet the amino acid profile or the essential amino acid score does not. So I think, this newest methodology of assessing protein quality, this digestible, indispensable amino acid score, I think it's very important for all of us to be familiar with it, to understand that it is going in the right direction in terms of looking at the bioavailability of amino acids and really being able to then apply specific age group related physiological requirements to that level of essential amino acid content and digestibility. And you get this, I think, great on a Venn diagram. This would be really that common shaded area. What foods contain, most of the essential amino acids, if not all? Which foods contain those in levels that are very digestible, and the word digestible meaning that the amino acids can be absorbed in the small intestine. And then how do those amino acid levels equate, or how do they relate, I should say, to the requirements of a specific age group? So I think this is the way of the future. The assumptions that are made with this is that we have the same digestive ability as a pig. There are some slight differences there. And another would be, again, that we just don't have the data that's necessary for such a large number of foods to this point. So, but it's going in the right direction, and it gives us, I think, a great confluence of some of the other tests together. Now, if we compare this DIAAS to the PDCAAS, and this is a great article. If you're not familiar with Sharon Palmer's article in Today's Dietitian, this is from the February 2017 issue. It's a phenomenal look at plant proteins in particular and how they can be combined, and the different, I guess you'd say, cost-benefit ratios to many of these plant proteins. And again, not just plant proteins. It does look at, dairy proteins, as you can see here, but you can see that the DIAAS for the milk or dairy proteins are typically higher than the PDCAAS. That's because, again, a DIAAS will go greater than 1.0. With a PDCAAS, 1.0 is as high as it can go. Whereas with a DIAAS, things can get up to the 1.59, which is the case with, certain dairy proteins. You can see that the soy protein, PDCAAS of 1.0, yet it only has a 0.91 for a DIAAS. So again, you'll see that there's significant differences, and a lot of this has to do with the PDCAAS's measurement of fecal losses versus the digestible indispensable amino acid score, which is looking at losses that occur prior to the distal end of the ileum. So it's a great article, though, for those of you who want to read more about this. Now, I've mentioned that there are several assumptions, and these are a few of them that I think it's important to remind ourselves of when we look at these. Really, I mean, most of these assessments have value. I wouldn't want to, say that any of these, really don't have a place in helping us assess a protein's value. It's they've all played an important role in that process over time. But we certainly need to know the essential amino acid content of a food. That's really important because those essential amino acids are required on a daily basis, by a human to support, growth, development, weight gain, etc. Then it's also very important to accurately measure growth, actual growth and or weight gain in an animal or a human model as it relates to the consumption of certain protein sources or protein-rich foods. I mean, I think that's the leap that sometimes is made with something like a chemical score, is that it's all theoretical, but we have to actually be able to see growth, whether that's in a clinical study or in our patients. Patients are going to respond differently to different protein sources, and a lot of that has to do with the specific amino acid content of certain foods, which we'll get to in a moment. And we have to certainly have some level of understanding digestibility of any given protein source. Having all the essential amino acids is important, but they also have to be bioavailable. And we know that there are a variety of factors in any individual or in any populations, in those characteristics that influence digestibility. That ranges from, pharmaceuticals that suppress, acid production, like proton pump inhibitors, they're going to certainly influence the digestibility of a protein, as well as overall stomach acid that often changes with age, levels of pancreatic or proteases secreted by the pancreas, the amount of surface area in the small intestine, all things we've talked about. So all of these things really must be taken into account, and that would make certain models or methods of assessing protein quality better than others. So with that being said, let's take a look now at some very unique roles that amino acids play. And, again, for the sake of this conversation today, we'll really just look at 3 or 4 of these. I think arginine is one that is most fascinating, largely because of arginine's unique role in circulation and in the production of nitric oxide, which, if you're not familiar with nitric oxide, it's really the primary method of vasodilation, getting blood vessels to increase in diameter to accommodate greater blood flow. And it's really important for everything from blood pressure to having a normal sex life, you name it. I mean, nitric oxide it's really tough to have quality of life without adequate amounts of it. It's obviously plays an important role in other aspects like our immune systems and our immune system's response to infection, which we'll look at more closely here in a moment. Methionine, the sulfur, one of the primary sulfur-containing amino acids, along with cysteine, that's really unique in the fact that it's critical to methylation. And we know that methylation plays, such an important role in neurological health, as well as in hepatobiliary health and our liver's ability to drive detoxification pathways or reactions. It's critical for that. And in so many populations suffer from inadequate levels of methylation, and those are the result of multifactorial issues. I mean, you have people with genetics where they're really compromised with respect to methylation, whether that's a MTHFR phenotype. That could also be with just inadequate levels of dietary sulfur, dietary methionine or cysteine, and so they don't have enough of this particular amino acid or the sulfur-containing amino acids to help with really the critical roles of those amino acids in methylation. So methionine is really it's fascinating. It's one that we won't spend as much time on today as we will arginine and leucine. Leucine is really the star player of the branch chain amino acids. And so when I say branch chain amino acids, I'm talking about not just leucine, but leucine, isoleucine, and valine. They have been deeply appreciated now for 50 or 60 years, especially in the area of sports medicine, because branched chain amino acids were shown, again, 50 years ago to really, really spare muscle, especially during intense exercise, during prolonged periods of stress. They have collectively a very strong anti-atrophy atrophic effect. So they've been, again, used intensely in the area of sports medicine, but, more contemporarily, they've been used in the critically ill in trying to, help those patients support more lean body mass or gain lean body mass because of some of the unique effects they have on our physiology. And then tryptophan, I think most people, whether it's a consumer, a patient, if you say tryptophan, they say turkey. And that is, the extent of knowledge that most people have about tryptophan. But as clinicians, we all understand its value in being a building block, so to speak, for neurotransmitters such as serotonin and others. And it's one of those amino acids where we definitely require, from person to person, individual to individual, we require different levels of tryptophan for optimal health. And some of that is, in its infancy, I guess, in terms of determining who needs more. But we certainly know that tryptophan can be used by patients who have major depressive disorder and have depression with varying levels of success or efficacy. So these are just a few of the amino acids, and in this case, essential amino acids that I wanted to give a little time to. So let's talk first about arginine. I mentioned that, arginine, obviously L-arginine, it's the L version of amino acids is really, that's what we're talking about here. Those are the ones that physiologically have the role in our bodies. Arginine, again, it's used to produce nitric oxide. And a lot of your foods that are very rich in L-arginine or in L-arginine's counterpart, and it's not a well-known amino acid, it's not required by human physiology, but it's closely related to arginine, is citrulline. Citrulline is found in foods such as zucchini or summer squash, and it's found in the seeds of every member of the Circurbita family, whether that be melons, like watermelons, in addition to cucumbers, that Circurbita family, the melon family, typically the richest sources of citrulline, especially the seeds of those. And that's why watermelon seeds, for instance, and zucchini, they've been referred to as poor man's Viagra, for at least two or three decades now. A lot of that has to do with the ability of citrulline, along with arginine, to drive nitric oxide production, to really improve blood flow, and that obviously has a lot to do with what Viagra does for men. So arginine, though, in addition to really driving this nitric oxide production and helping with circulation, it appears to be an amino acid that we need to focus on more for the critically ill patient, especially patients who have dealt with, more long-term systemic infections. A lot of this has to do with the body's oxidation of arginine during periods of illness or infection, and it can create, much, much greater metabolic demands for arginine than would be, I think, normally expected. And so I've got a slide towards the end of today's talk that will look at that. So arginine is something that really should be well represented. And I know that I mentioned earlier in this talk that it was a conditionally essential amino acid, but I would argue that it is more often essential than conditionally essential. And a lot of that has to do, again, with, the situation that any individual or population may be within. But I would say when you take a look at the lifespan, the needs of arginine over the course of the lifespan, and looking at, how many people, we really now have in the world that are over the age of 50, which is where that arginine requirements start to come really back into play. So it's not just adolescence. I think we need to really look at arginine requirements during our older years, later in life. I think it's an essential amino acid more than conditionally essential. But again, that is more of my opinion than a than a consensus across the world of amino acid research, and if you talk to amino acid experts. That being said, these are your best sources of arginine, those which we're going to deliver the most arginine per gram of protein. Pumpkin seeds are phenomenal. From the animal world, poultry and dairy products, whether that be milk, yogurt, cottage cheese. I mean, cheese in general is very concentrated, but pumpkin seeds are one of the best from the plant world, along with peas, green peas, lentils, chickpeas, and soybeans. All of these are excellent, and nuts and seeds can be pretty decent sources as well. Now, I mentioned methionine earlier. Again, as a sulfur-containing amino acid, we really know typically where methionine and cysteine are found, because if you've ever smelled, spoiled milk, you can really smell the sulfur starting to come off of that milk as it spoils, or there's bacterial microbial activity. But it's, methionine is most concentrated in animal protein. Foods such as your legumes, like white beans and soybeans, they do have pretty decent amounts. Brazil nuts are unique in their concentration, and so is quinoa. But overall, dairy products, meat, poultry, those are where you get the most methionine. And usually methionine comes into play when you start talking about plant proteins not providing all essential amino acids. This becomes, along with lysine, this becomes one of the more difficult amino acids to get if you were going to just pick one plant food and try to run with it and use it as a sole source of protein. Usually it's either methionine or lysine that becomes the rate limiting amino acid. If it's not one of those two, then the third would be, typically it's tryptophan. But so that's why this is an important one to to understand, not just for its role in methylation and its ability to help us detoxify substances. A lot of it also, its importance is the fact that you want to make sure, if you're only eating a plant-based diet, that you've got sources of methionine, whether it's quinoa or it's a type of legume present to go with, the other plant sources that would not otherwise be providing it. And, again, this is a great illustration of its role in the methylation kind of homocysteine conversion pathway. And a lot of us are familiar with homocysteine as a biomarker of potential cardiovascular issues. We know that, for a long time now, that higher levels of homocysteine can be associated with different issues, cardiovascular, neurological, cognitive impairment. I've had so many observations from my clinical experience over the years where higher levels of homocysteine were present as an individual was really suffering from a variety of symptoms. So it's, I think it's a great biomarker to look at in patients. We don't always get a chance to do that clinically, but a lot of that homocysteine value as a biomarker, its ability to reflect how much methylation is going on in a patient, and whether it's choline or betaine, or I mentioned glycine earlier although it's a non-essential amino acid, it is pivotal in this methylation pathway. But methionine is front and center. And so again, a credit to Chris Masterjohn, who just does great work. If you're not familiar with Chris's work, I really encourage you to take a look at some of the material that he's written over the years. So again, methionine is important. It's important for, much more than protein synthesis. It really gets into critical pathways in the body. Now, lastly, here, before I, take a moment to answer whatever questions that you might have, I want to conclude the kind of the amino acid look with leucine, because leucine, as I mentioned earlier, as a primary kind of star player amongst the three branch chain amino acids, leucine has the ability to not only increase muscle protein synthesis in a unique way, meaning much more so than just the value of a typical essential amino acid. It's been shown that when you get to around 3 or 4 grams of leucine in a meal, that muscle protein synthesis takes a big jump, meaning that there's a much greater gain in muscle protein synthesis than there would be with another particular food that had all essential amino acids, had a great PDCAAS score, but didn't have as much of let's say, leucine or a leucine content overall. So at 3 to 4 grams per meal appears to be, from the most recent research, that's that area where you start to get the real exceptional lean body mass gains, muscle growth in a unique way related to leucine. And a lot of this has to do with its ability to inhibit myostatin. And myostatin is something that, usually we want to have that in homeostasis, with other enzymes that are driving protein synthesis. Myostatin is something that really is responsible for muscle catabolism, and it really comes into play during long-term endurance type exercise, where you get, the catabolism of skeletal muscle to meet, basically to meet energy demands by the body. And, myostatin can be inhibited with higher levels of leucine. So that's really important for our critically ill patients that have had significant losses of lean body mass. And that's why I think it's so important to concentrate on, rich sources of leucine for those patients. Here's a look at some of the, animal and plant sources and the leucine content that they contain. You can see that, your dairy is at the top of the list with respect to, especially whey protein, where you lose, you have a higher percentage of branched chain amino acids in whey than you do in casein. So when you fraction that out and you separate the whey and the casein, you get even more of those branched chain amino acids. So that's why whey protein is considered to be the gold standard or best overall source of leucine. But that doesn't mean that, again, the plant sources can have adequate amounts of leucine. And you can, in fact, as you'll see here, you can get 3 grams of leucine with something, like, pea protein. You just need around 39, 40 grams of it. You need a little more of other types of protein. You need a little less with, rice. So again, pea and rice protein, soy protein, they have the leucine there. You just need to eat more of them than you would, let's say, whey, approximately twice as much. But that, again, there's other things that are found in pea protein. So it can really serve well with respect to helping meet leucine needs. So now, for just the last few minutes, I want to really give some attention to the protein quality considerations for the critically ill. As I mentioned earlier, again, the notion that plant proteins, complementary plant proteins, can be consumed over large windows of time to meet an individual's overall essential amino acid or protein requirements, that may be true for, healthy young adults. But it is certainly not the case for older adults or the critically ill. Those individuals or those populations must consume these complementary plant proteins, or these, different sources of essential amino acids, in very small windows of time, i.e. the same meal. I think that's a really important consideration for the critically ill patient. The second is that a loss of lean body mass often requires higher levels of branched chain amino acids, and most specifically, or I should say more specifically than isoleucine and valine, would be the role of leucine in really helping these type of cachexic or extensive catabolic patients, those with a higher metabolic requirements, helping them have better outcomes. It's important to really focus on the leucine content. And then it's important to recognize the role of L-arginine and how much of that is used with long-term infections and illnesses. And we need to keep that on our radar when we're dealing with patients who have had, long-term infection, significant, windows of time with illness. And you would think that they were getting all of the essential amino acids that they require, but we're not maybe seeing that in their weight gain or lack of weight gain. They're continuing to lose weight, or they can't gain weight. This may be responsible for some of that. And, again, this is a great paper, Nutrition and Clinical Practice, that 2017, one that I really like, that looks at this, which shows how amino acid oxidation, can really start to, this is the oxidation, which means that amino acids aren't being used for the body's overall protein pool for protein synthesis. They're being oxidized at a much, much higher rate in the critically ill patient, and that's where L-arginine and others like leucine need to be really taken into account and looked at closely. And this really speaks to that, looking at how quickly arginine becomes the rate-limiting amino acid. Out of all the essential amino acids, it can often become the rate-limiting amino acid, despite what we think is present in a particular formula, enteral formula, or in their nutritional support. So again, most of our attention would be on something like lysine as a rate-limiting amino acid because of how it appears in lower concentrations in plant foods, but L-arginine can quickly become that pivotal role when we're dealing with sustained illness or infection. So again, I want to just say that it's important to really assess changes in fat-free mass as opposed to total weight, because often we measure in a patient we measure weight gain, we measure weight loss, and we inadvertently bring into that measurement a significant amount of fat mass. Body fat mass is not something that we really need to assess when we're trying to determine changes in lean body mass. So when we're assessing our patient's response to a particular diet, a particular type of formula, or any type of nutritional support, we really need to, if we can, kind of pull these two assessments apart, which is fat-free mass, which we call lean body mass, and body fat mass, really separate those so we get a better picture of what's going on. And so in summary, patient assessment really needs to be made upon several factors in determining protein quantity, and to today's conversation, protein qualities, age, right? The amount of lean body mass that's been lost, what's there with respect to, surgical, burn type trauma, do we have a large, area of the patient's body or significant percentage of their lean body mass that's been compromised with surgery or wounds? Is there insulin resistance present? We know all of this can really influence protein requirements, both on a quantitative and a qualitative level. I think as clinicians, we really need to observe a patient's response to both the amount of protein that they are consumed or given, as well as the sources of protein that are playing, primary protein sources in their diet. We have to always appreciate tolerance and digestive function, because if they can't tolerate it and they can't digest it, then it doesn't matter, what the PDCAAS score was. If there's the presence of something like an allergen, or it's really, changing transit time dramatically. As I just mentioned, looking at lean body mass versus overall body weight, it's much more helpful to look at lean body mass changes. And I think we need to really look at the quantity of protein given at each meal. We know that plays a much bigger role than, let's say, the quantity at the end of the day. We need to look at the daily amount, but again, the sources of protein that are present both at each meal and at the end of the day. And so with that in mind, we know that specific populations can have very unique metabolic requirements that could alter specific amino acid needs. We know that lean body mass changes, really determine the optimal amount of protein and the role of specific amino acids, as is the case with leucine. Digestibility is critical. Plant proteins will always require multiple components to acquire a higher biological value, or, again, more contemporarily here, this digestible, indispensable amino acid score. The current RDA of 0.8 grams per kilogram of body weight may not be adequate for many patients. They may require 2 times that amount for an optimal recovery. Leucine and arginine are important for long-term illness, loss of lean body mass, and that, again, in conclusion, the assessment of dietary protein quantity and quality and the overall effectiveness of any particular source of protein needs to be made on several different observational measurement levels, but ultimately, looking at how the individual responds in terms of various facets of their physiology, with lean body mass, being a big part of that, That should always be our gold standard. And so with that, I want to thank you for your interest in this topic today. I want to thank Nutritional Medicinals for, this forum to really cover this type of material, and I'll do my best to answer whatever questions you have. Thank you.
Great. Thank you so much, Dr. Bagnulo. So before we dig into the question and answers, I just want to let everybody know that this was approved for one CEU. So by Friday, you should have an email from us with your CEU certificate and references for this webinar. And then within the next 1 or 2 weeks, you should also be able to see the recording on our website. If for some reason you do not get the email with the certificate and you've checked your junk or spam folder, please reach out to us, or you can reach out to me directly at laura@functionalformularies.com, and we will get that to you as soon as possible. So a few questions, and I don't know if we'll be able to get to all of these, but we'll get to as many as we can. Do you recommend branched chain amino acids for the average person who is beginning to incorporate exercise with the goal of weight loss?
Laura, was that the final part of that with the goal of weight loss?
Yes.
Not necessarily. No, I think branched chain amino acids are more valuable to the individual that is involved in a high level of training and is trying to prevent muscle loss. So I would not look at branched chain amino acids as being as useful for someone that is incorporating more exercise and really looking for weight loss. I really look at it more for weight maintenance or weight gain. The other, the other population that does appear to have a really favorable response to branched chain amino acids would be the ketogenic population, clinically, or those that are trying to apply a ketogenic diet for other health benefits in their day-to-day life. And I know if you want to speak to that, Laura, obviously as an expert in the area of ketogenic therapy, I've seen people have really favorable response to higher levels of branched chain amino acids when they're on a ketogenic diet.
Yeah, I think there hasn't been very much actual research done on that. It's like in its infancy, but the few papers that have come out on it certainly show favorable effects of boosting ketones while also providing, a protein source. So I think it's definitely something that research is expanding on for sure. A few questions here about arginine specifically, Dr. Bagnulo. So can you please talk about the role of arginine specifically in wound healing? And then the second part of this question, what supplements would you recommend for wound healing that contain high arginine content, or maybe what food sources in terms of the wound healing aspect?
Yeah, sure. It's a great question. I think that it definitely plays a role in improved wound healing and improving outcomes for those patients. For a long time now, even back in graduate school, I was fortunate enough to be involved at that kind of entry level with research on the arginine content of an animal's diet and wound healing. And it was really clear then, 30 years ago, that higher levels of arginine definitely facilitated faster or more rapid wound healing. A lot of that has to do with just kind of the microvessel circulatory delivery of nutrients to a wound. Blood flow is really important to a wound to deliver everything that's required to get wound closure, to get normal protein synthesis there. You have to have blood flow get there. So I am a big fan of arginine-rich foods. I think pumpkin seed protein would be one of my probably be my first choice from a plant source, and if it's not pumpkin seed protein, then I would say whey protein isolate would be another excellent source of L-arginine. Typically I don't recommend that individuals take amino acids on their own. I know that certainly that's not the consensus. There are many clinicians that recommend giving patients amino acids on their own or in isolated form. The only reason I have reservations around that is I think it is easier to develop amino acid imbalances once that becomes the approach. I think if you use a whole food, or at least the protein content of a food, something like whether it's pumpkin seed protein or hydrolyzed pea protein or whey protein, that they may not be entirely whole. I understand that they're somewhat fractionated, but you're still getting more of that symphony of amino acids and nutrients. You're going to be getting all of the L-arginine there. You're going to get it in high concentrations, but you're also going to get other amino acids and other nutrients that overall play at least some role in what I like to refer to as food synergy. So I tend to recommend pumpkin seed protein or whey protein as better sources of L-arginine. And honestly, in the research that I was involved in, again, this was going back 30 years ago, but I've seen other papers that, I feel mirrored this, is that L-arginine, whether it's delivered from a food or it's delivered in isolated form as an amino acid, as long as the L-arginine is there, that's when you get the improved outcome. So I don't think it's a mandate for using L-arginine on its own as a supplement.
Okay, great. And speaking further with arginine, is arginine still bioavailable for, like, say, pumpkin seed that has been roasted? Is there any impact on cooking and the bioavailability of it?
Actually, cooking enhances the bioavailability of it. L-arginine is going to be more bioavailable in a food that's been roasted or boiled or cooked than one that's raw. Again, it's not to say that that we shouldn't eat those foods raw. Like raw pumpkin seeds is a very healthy snack, but the cooking process would enhance the bioavailability of it.
Okay, great. I know we're over on time. We'll answer 2 more questions, but if you have to log out, we totally understand. This question is, and I really like this question. We generally do not eat foods in isolation. So, you just don't eat protein all by itself. So is there any food combination that affects protein function more than others? So in terms of carbs and fat, like, are there ideal combinations with those macronutrients?
That's a great question. I agree. We don't eat one particular food on its own. However, as I mentioned earlier, that was, what was certainly done with some of those, protein efficiency ratios and biological values. They're looking at an animal's response to a food eaten alone. And I know that we don't do that. The thing I would say is that protein, protein digestion and protein metabolism overall, which is everything that goes into the digestion, breakdown of, and then ultimately the assimilation of it. It's influenced by so many different facets of physiology. I mentioned, for instance, in our last discussion that we had on protein quantity, how insulin can really change the way our bodies use protein and amino acids. And I would say that there is with respect to insulin in particular, we have this a bell-shaped curve. So meaning that if you have a very small amount of insulin present, it's going to facilitate amino acid uptake of any given protein, and it's going to really help with delivering those amino acids to muscle cells or to any other compartment of our what we call lean body mass, and increasing protein delivery and use there. But then once we get kind of over the top of that bell-shaped curve, that's when there's that, not only do you end up in a place of lower diminishing returns, but it can start to compromise the way our bodies are ultimately metabolizing and utilizing protein. So, for that, the purpose of that would say that, if we had protein, not on its own, but we had it with a, very modest amount of a healthier form of carbohydrate, whether that be something like, whether it's potato or sweet potato or, some type of, starchy root vegetable. Those are always my first, because of just all the nutrition that they bring to a patient. If we had something like fish with a sweet potato, or we had any protein source that was with one of these starchy root vegetables, carrots, beets, things like that, then the modest amount of carbohydrate present in those root vegetables is enough to stimulate a small amount of insulin production, which is ultimately going to facilitate greater protein acquisition by a muscle cell and therefore use and efficiency. So I don't know if that helps, but I do agree. We don't typically eat foods alone, although sometimes I guess we do in the case of some of those dairy foods that we covered today. and someone might snack on something like pumpkin seeds on their own. However, I do think there's value. Again, I really like this concept of food synergy. If you have protein with a healthy source of carbohydrate, such as a starchy root vegetable, that I think, the way the body uses that protein can be more effective, more efficient than if it was eaten on its own.
Great, thank you so much. So I know we're 5 minutes over, so I know there were some questions we didn't get to, and we apologize for that, but Dr. Bagnulo, we really appreciate you for spending all this time with us today. And if your question didn't get answered, please feel free to email me. Again, I'm here to help, and I'm happy to try and get those answered as much as possible. So thank you to everybody for attending, and we wish you the best in health.
Thank you, Laura.